Okay. Check all my places. No questions there, no questions here, no chats. All right. So this, I think, is a nice ending topic for the semester. We talked about how phenotypes can be inherited by the sort of introductory Mendelian allele inheritance conversation about basically you get something from your parents and it turns into your phenotype. And then we talked about molecular genetics, where we talk about the DNA being the thing that got inherited, and then that becomes your phenotype. And then we understand how DNA causes phenotypes by causing proteins to be made, and those proteins have functions, and those functions give you maybe a different phenotype than another individual of your species. And so this final topic is really an important component to genetics, and a lot of people, when they're researching phenotypes, they dive right into this level of research where they want to know, okay, well, we have this instruction manual for how to make a person. You've got your genome and all your instructions for your genes, but how and how often and where in the organism, how do those genes actually get turned on? Are they all on at the same time? Are they on at different times? And that's the subject of gene expression, as we'll talk about today. Kind of one of the things we were talking about in a previous previous lecture, I thought this was maybe an interesting thing to compare. This is a comparison. They've got a lot of DNA data comparing humans and our living and extinct relatives. So humans have related species that have gone extinct on the path toward making the modern human, and then we have living relatives in these apes, the chimpanzee, orangutan, and so on. And this was an interesting study where it's basically a mutation that turned a gene back on that caused this to be an important component to why we have our big human brains. So a little bit on the subject of DNA that might be useful, might not be useful. This was a situation where this gene sequence, you can see this human version and primate version, have similar DNA sequences, but based on the mutation, there's actually a frame shift mutation here that causes these different, these single letters that are more spaced out on the bottom. Those are the amino acids that correspond. So by shifting the frame of this protein, it either has this important function in humans, or it has a basically non-functional version in our living primate relatives. And so you can see that kind of important changes can happen as a result of frame shifts. Okay. So the subject of gene expression, if you want to think about your DNA as a library, I think that's a useful way to think about it. So you've got this DNA genome, which contains a whole bunch of instructions for putting your proteins together, other components of yourself, your ribosome, your tRNAs, and so on. And a library is full of books, but people aren't reading all those books all the time, right? So it's information there. If you needed to access it, it's there, but it's not always being accessed. And the same goes true for your genome, that you have some instructions in the genome that might not be turned into proteins all the time, or even very rarely turned into proteins, even though you've got instructions for how to make those proteins. And this is important because, if you just took all the gene instructions that you have and just made them all into proteins all the time, that would make like all of your cells would be uniform, and your cellular protein components would be the same at all stages of life, across all different organs in your body, and so on. And that's not a very adaptive way to live. So instead, there are mechanisms that we'll talk about with this gene expression conversation, mechanisms that turn some genes on in response to environmental signals, developmental signals, other activity of other genes, and so on. So we're going to kind of start small. The first ones we talk about on this topic are going to be bacteria. Bacteria have typically smaller genomes and are less complex. So they're cellular organisms without organelles, and they do things a little more simply. So starting with one of the most widely used bacteria, this is one of the model organisms for studying bacteria. Our friend E. coli has, in one version, 5,416 genes. But which one of those are being transcribed at any one time? It's not all of them. So it's a subset of those. And this I found to be an interesting study from a couple of years back. Scientists were interested, if you take one of these small genome organisms, like one of these bacteria, all right, so it's got 5,000 genes. What if we took away some of those? How small can you get a bacterial genome and still get a functioning organism? So these researchers basically took bacteria and took away one gene at a time and said, all right, we'll take this away. Can this organism still be alive? Keep in mind, of course, this is an organism that's being basically pampered. So it's grown on full nutrient medium. So it has all it needs for nutrients. It has no competition from other organisms. It has nothing trying to eat it, no viruses trying to attack it. It's perfectly grown in isolation in wonderful conditions. And under those conditions, they could strip down this bacteria to the point where they got only 473 genes. Sort of a minimal size bacterium. So that 473, that would, if you think about the function of those would be really essential genes, right? So genes that this organism can't do without under any circumstances, even when the cell has everything it could possibly need. So what do you think would be some of the most important genes to have you couldn't possibly do without? Turn this one off because I won't be looking at it. You can chat your answer if you like. You could turn on a microphone and flex your vocal cords. What would you think is an essential gene function? Nobody? There we go. There's one. All right, growth. So all right, so what does it take to do growth? Growth is kind of a broad concept. So to grow, we had to make new stuff. So make a new cell. New cell has proteins, lipids, DNA. So structural components, right? Maybe if you're looking at a bacteria, cell wall, something like that. And of course, if you're living on a food source, we need the genes responsible for taking up the nutrients to make these things, right? So if you're a cell and you want to make new cells and grow, then you got to take in nutrients convert those into the molecules that you need to build new cells, all the enzymes that are responsible for building those, organizing everything. So kind of cellular, I'll go ahead and put a term in here that we'll see a little bit later. Housekeeping. This is a term that scientists use when they talk about these sort of baseline functions that are responsible for basically keeping cells alive at a really basic basal level. And a lot of those are also related to central dogma. So we've got processes of transcription, translation. And there are enzymes responsible for these processes. So those enzymes need to stay on if this cell is going to be alive and make copies of itself. Maybe while we're talking about this, I don't know what to call the other ones. We'll call them elective or maybe luxury functions. So if we got essential functions, what would be some of the functions that maybe a pampered cell can do without? All right, quiet morning, huh? So I mentioned that the study involved in growing these bacteria on media where they're pretty well taken care of. So this would be cells that don't have to, say, defend themselves. I don't know, what do you call something that eats bacteria? We'll call it a predator. So maybe some defense pathways, molecules that might be involved in defending against viruses or competitors. There is, right, as Colin suggested here, some communication between cells. You can have cells of the same species, sometimes communicating through chemical signals. There are also bacteria that sort of communicate with each other through chemical signals. So if you're the only bacterial species, it's not really required for you to partner with anybody else, communicate with them. This list would go pretty big if we thought about all the different ways that cells basically are equipped to respond to rare situations or uncommon situations, but not essential situations. One of the things we'll see in gene expression is responding to the presence of, say, a new food source. So something is available in your environment and you need to have an enzyme that can break it down. A lot of bacteria are equipped to make use of different molecules they find in their environment. In the research study, the researchers are providing them basically with just straight up sugar and protein amino acid building blocks. And they don't have to get those for themselves, but a bacteria in the environment might need to make use of rare things, molecules that it finds. So it's sort of a basic, essential metabolism, and then this extra layer of optional, sometimes used metabolism. So cells have those genes that aren't always being used, but they can turn them on. And this is the process of gene expression. So if we take central dogma here and just strip it down, when we talk about gene expression, we're really just going for this process. So scientists who study cells and DNA they understand, of course, central dogma, and they don't have to talk about it or think about it all the time. A lot of times we're interested to know, okay, what's being made? What proteins are being made at this particular moment? And that's the process of gene expression. So if we skip out the middleman and just ask the question, okay, is this gene on or off? And so we have a little bit of how I frame this topic is we just think about these genes as having switches that turn them on or off. So if the gene is on, protein is made. If the gene is off, the gene is still there, but the protein doesn't get made from it. That make sense? Okay. So let's say you had a gene. How would you cause it to be expressed or not expressed? Prevent it from getting copied. So like DNA replication? Yes. So DNA replication is a little bit different from gene expression. If a cell loses the DNA for a gene, so are you suggesting maybe that we make a copy that's missing a gene? If that happened, if you say failed to copy one of the genes, then that would really prevent this cell or any cells that result from it from having that gene or ever using them again. So that would be a little more extreme version of turning off a gene. If we think about the process of turning a gene into a protein, there's a little bit that has to happen to get there, right? So along the way from having a gene, the DNA, and turning that into a protein, transcription has to happen, translation has to happen, and if you disable one of these, one or the other of those, then that really prevents this process from going forward, right? So you can stop a gene from being transcribed, translated, or as we'll see, some other steps in this process. If you can just prevent that specific to a gene, prevent that process from happening, then that gene's protein doesn't get made. So here's my silly analysis here. You take Batman. Batman is let's say there's a gene containing instructions for Batman. Batman comes into play when the commissioner goes out and puts up the bat signal, right? Turn on the bat signal, Batman shows up. Batman is always around. Sometimes he's doing laundry. And so it takes that signal, in my simplified premise, takes that bat signal to get Batman to get in and do what he does as Batman. So if you turned off the signal, let's say you disabled the bat signal, then maybe Batman would never be called into action. How's that for an analogy? Okay, so this is from your book, just a little more specific version of this. Basically, we're going from the DNA that's present in the genome all the way to a functional active protein. And this shows some processes that are specific to eukaryotes. But in general, this going from DNA to protein, this is the process of gene expression, and this happens in bacteria or eukaryotes, at least the start to finish parts. And all these areas that are in the yellow bars, those could be affected in a way that would prevent you from getting to the finish line. And if you don't get to the finish line, the protein isn't made and the protein doesn't do its job. right? So I have a couple arrows on here for over here. This is transcription is one of the more common ways we think about gene expression, and we'll see why that is as we go along. So transcription is an early step in the process, and it's a process that can be affected a little more specifically based on structures of promoters and so on. But it's also possible to affect how RNA is processed, RNA stability, whether that molecule sticks around long enough to be translated, modifications of the protein as it becomes active. So keep in mind transcription, we're going to spend a lot of our time talking about that as a level of regulation. And then, of course, some of these, anything having to do with splicing out introns, adding this 5' cap and so on, those are specific to eukaryotes, as we've seen. And just a little throwback to lab. One of our labs, we did the RNA interference. So what step would RNA interference affect in this process? Well, it's got RNA in the name. So it's actually realizing I don't know which one of these it is, but it's one of these where we have an RNA molecule. So the RNA interference, if you remember, was where cells can be sort of tricked into attacking their own RNA. So we added this double-stranded version of genes, and that caused enzymes to go in and destroy one of these versions of the green RNA molecule. And again, that prevents this process from moving forward. If you destroy the RNA, there's no instructions to be translated into a protein, therefore the protein doesn't get made. And that RNA interference tool that we used in lab is a very powerful tool to specifically take out one kind of RNA, the RNA corresponding to one gene or one protein. You can take that out systematically in a lab environment. And that's what we did with the roundworms last time in lab a couple of weeks ago. We gave them RNA, double-stranded RNA, to turn off specific genes, and we got to see the results of that, right? Kind of coming back around with this slide to something we talked about already, those genes that are a little more essential. So there are genes that their instructions are essential, and then there are also genes that are basically always transcribed. So if you think about maybe the function of people working in a city, right? Some people can take vacation and go home, but the people who keep the lights on and keep the water running, keep the, I don't know, keep the food being delivered, these important functions that the city couldn't do without, those people, their jobs are always essential. Other ones are more luxury or extraneous jobs. And same thing goes for cells and genes. They're genes that are basically always transcribed because they're always needed. And these are related to those really baseline functions. So this is from a study. What this shows is a set of genes that they use to look for the presence of different kinds of bacteria in the environment. And what they're doing, they're looking for RNA from these organisms. And so if they want to find the RNA reliably, so you're basically like digging up a spoonful of soil and trying to find evidence of organisms in there without finding the organisms themselves. They take those samples from the environment, soil or water, and they can look for RNA from specific genes. And so if you need to find an RNA for an organism, you want to find the RNA that's always made. And that's these so-called housekeeping genes, or a fancier name for those is constitutive. And both of those mean basically genes that are always on, and you should always be able to find the RNA for those in an organism. So what kinds are they? I'm not sure of the functions of all of these, but a big block of these RPS and RPL, are codes for ribosomal protein of the small subunit, ribosomal protein of the large subunit. So these are proteins involved in making the ribosome. And of course, the ribosome involved in translation. If a cell was ever not able to do translation, that would be pretty horrible for the cell. Cells have to translate RNA messages pretty much all the time, right? So if you looked for an organism and you were looking for its RNA for a non-essential luxury gene, you might not find it because it just be made at that time, not because the organism isn't there. All right, good. Cool. Okay, so always on, these are basically always being translated. Other ones, their translation, transcribed and then translated. Sorry, focusing on transcription. Always transcribed, and some of them are not always transcribed. So the level of transcription here. And this is a diagram. We're going to see a lot of variations on this diagram, so get familiar with it. What it shows is the red region here would be a promoter. And this green molecule here is responsible for enabling transcription to happen. So this green molecule gets in there and allows this enzyme to do transcription. I flashed it up on there, but I also have a question about it. What is the enzyme responsible for transcription, the big one? That's right. All right. Oh, DNA polymerase. So transcription would be RNA polymerase, right? So it makes the RNA molecule as a result of transcription. So RNA polymerase gets in there, does transcription. And what we're going to, we've kind of hinted at this before, but we're going to spend more time talking about what this green molecule is and how it affects the process of transcription. So before we saw transcription factors, right? So say we've got our gene here. So we saw a situation where there were transcription factors. And they basically attach to the promoter region, and they set up an environment where RNA polymerase is then sort of invited to the party, right? So the transcription factors are important for kind of being the middle enzymes in between the raw DNA sequence and then getting this RNA polymerase in there. And so the question of gene expression is maybe these transcription factors are not always attaching to the promoter. So the RNA polymerase is ready to do transcription at places where it's invited, but if the sort of entourage of enzymes, if they're not available, then RNA polymerase might not get the signal to do transcription. Okay, so just a bigger version of the same diagram here. This green enzyme comes in, and in the form that it's in, it attaches to DNA and enables the RNA polymerase to come to the promoter causes transcription to happen. However, there's another version of this molecule, this activator molecule, where it can be inhibited and prevented from attaching to the promoter. So this would be an inactive activator, can't attach to the promoter, and if it's not there, RNA polymerase doesn't recognize this as a gene that needs transcribing, and it doesn't transcribe. And here we've got this on-off situation. So we're going to see a few versions of this where genes are on, genes are off. And this is one way that transcription can be turned on or off, is through the action of an activator. And the activator is a form of transcription factor. So we have molecules called activators. And repressors. And they're in this category of molecules that are transcription factors. They recognize DNA at specific locations and cause, or sometimes, as we'll see, they can prevent transcription. All right, still with us? All right, so we've seen this, proteins that are not always required. Some molecules, some transcription is what we call induced, and some is what we call repressed. So we're going to see a whole bunch. Unfortunately, this topic, the whole topic of gene expression in bacteria, has a lot of like sort of two sides of the coin ways to look at things. So we have already activators and repressors. And we're also going to see genes that are induced or repressed. So if a gene is induced, we're going to think about that as being usually off. But it turns on in the presence of a signal molecule. Right, so to induce something is to cause it to happen. And the way we're going to use this term inducing a gene is that the gene transcription is not on until the signal molecule comes into play and causes it to turn on. So we'll see some examples of these. We're going to see basically all kinds of variations in the way that genes can be usually on or usually off, turned on, turned off, turned on by activators, turned on by repressors. We're going to see the whole set. And unfortunately, it's a little bit to keep track of, but it's a nice, I think once you get the perspective for how this all works, I think it all fits together pretty nicely. So one more thing is being added by this diagram here. We don't talk about it too much, but there's some times when we will talk about it. This transcription factor, in this case the activator, is itself an enzyme, right? So there is actually a gene for making the enzyme that does the transcription activating. So that's what's being shown here. This could be near to where the acted upon gene is located. It could be at a whole different part of the genome. They don't have to be connected in any physical way. So that's what this dashed piece of DNA is supposed to indicate. So the activator is made. The activator enzyme attaches to the promoter, allows RNA polymerase to come in here, turns on transcription. And then another thing that's being added in this diagram is that basically the enzyme produced by the gene also causes something to happen. So if you want to get like all the way to phenotype, the ability to do a process is sort of the phenotype of the gene. The result of expressing the gene is that this enzyme process happens. And we're going to kind of simplify this down a little bit, where we're just going to talk about a single molecule conversion. So one little biochemical switch. And in this reaction, as it shows, we're just going to use the terms substrate and product. And I've had a little bit of students, they push back on this a little bit. I don't know how else to call it. Substrate can be tricky because we're going to look at different kinds of enzymes, and these enzymes each can have their own substrate. We're interested, when we use this term substrate, to talk about the molecule that the enzyme produced by transcription acts upon. So hopefully that's okay with you. So substrate converted into product, and this reaction only happens if the enzyme is present, and that has to be present when transcription happens, right? Whew, time for some examples, huh? Okay, so here's some cases of inducible expression. And I've got some crosses over the terms that your book uses. There's already a lot to keep track of with inducible, repressible, activator, repressor. So I didn't want to give you one more to worry about, this idea of negative inducible or positive inducible. I haven't really run into that in my experience looking at gene expression, so it seems to me one level of vocabulary you probably don't have to worry about. So we're not going to name them as terms, but we are going to talk about what happens. What's the common theme here is that both of these panels on the left and on the right, they both show genes that are turned on. And so again, that's the process of them being inducible, right? So there can be different ways that they are inducible. We haven't seen repressors yet, so this is the first time we're going to look at repressors and also activators together. All right, buckle up. So a repressor molecule is a enzyme that prevents transcription when it's allowed to attach to the promoter. So this is a molecule that actually prevents RNA polymerase from attaching to the promoter. Not something we've really talked about before. So it basically sits down on the promoter and attaches to the DNA and prevents that RNA polymerase from getting in there. And if the RNA polymerase can't get in, then transcription can't happen. So this repressor, in its active form, prevents transcription. However, in the presence of the round molecule here, circle molecule is the substrate. In the presence of the substrate, then the repressor changes its shape and now cannot attach to DNA. And in this case, if it can't attach to prevent transcription, then transcription will happen. All right. We've already seen the example on the right. This is an activator. The activator, when it attaches to DNA, allows RNA polymerase to attach, allows transcription to happen. And in this case, this is an inducible system where it's inducible using an activator. So in its form without the substrate, the activator cannot attach to the promoter. When the substrate is present, it attaches to the activator, causes it to change shape. In its new shape, it can attach to the promoter and allow transcription to happen. So the end result is the same in both of these situations. The presence of the substrate allows transcription to happen. Presence of the substrate allows transcription to happen. And two different ways, one using a repressor on the left, one using an activator on the right. All right, so let's take a little break and diagram this one. So basically, I've already talked about inducible when I talked about genes being induced. I think it's a little helpful to think about this inducible part. So inducible means that it's not usually on, but it can be turned on. So it's able to be induced. So gene usually off. Turns on in the presence of a substrate. So in this whole topic, we give you some pretty simplified versions, and in the simplified versions of these, the molecule that turns transcription on, in our cases, is going to be a substrate. It's a lot to keep in your brain, and I don't want to make it too complicated, so we're going to keep it simple. When we talk about substrate, so I'll use the same shapes that your book had. When we think about this reaction of converting substrate into product, it's basically the process of finding a food source or responding to the presence of a food source. Let's say it's a rare food source. This is the cellular equivalent of getting your salad fork, I guess, or what's a rare food? A lobster fork. So you find yourself with lobster, right? Time to eat lobster. Can't eat the lobster straight away. You got to get your special lobster eating equipment. So a cell finds itself with say a rare food source, a molecule that it can break down and use for energy. Because it's a rare food source, it doesn't make sense to have the enzyme always present. You don't always have your lobster fork out. So the cell responds to the presence of the food by making the enzyme that can break it down. And the end result of that is the cell gets energy from it, right? Is there a lobster fork? Picturing the one with the little one, you dip your lobster in the butter. Hopefully nobody hates lobster. Maybe we'll stick with this a little bit later, a little bit more. So still on the subject of inducible genes, we saw cases where the gene can be inducible using a repressor. if the repressor is active, able to repress, then it's able to attach to the DNA here and prevent transcription. And if the repressor is inactive, that allows the gene to be on. So if we prevent repressing, then the gene can turn on. And this whole process can happen also using an activator. That's the right-hand side of this diagram. So an inactive activator is what has the gene off. And an active activator allows the gene to be on. If you want a different way to think about this, I like to simplify these sometimes. If you think about them as just like positives and negatives, so active is a positive, repressing is a negative, then if you multiply a positive by a negative, you get a negative number, right? Back to algebra class. You have an inactive or a minus on a repressor, which is a minus, then minus times minus is a plus, right? Multiply a negative number by a negative number, you get a positive number. So inactivating the repressor gets you the gene turned on. So plus for gene on, minus for gene off. We could do this also with the activator. The activator is more of a positive thing. When it attaches to DNA, causes transcription to turn on. So inactive activator is a minus times a plus, that's a minus. An active activator is a plus times a plus, that's a plus. Right, so we can get this outcome of transcription on or transcription off using a repressor or using an activator. Still with me? Okay, so inducible, thanks. Inducible, and then repressible. So repressible genes are genes that are usually on or on in the absence of this signal molecule. But when the signal molecule is present, this is now the product of that reaction. When the product is present, then that turns transcription off. So these are repressible. The signal molecule interacts with the, in this case, with the repressor and activates the repressor, causes transcription to turn off. The product signal molecule interacts with the activator, turns the activator off, prevents transcription. So again, the key here is how much of this diamond product molecule is present. If there's not a lot of it, transcription is on. If there's a lot of this, if it accumulates, then transcription is off. All right, so that was a block of inducible. We can look now at repressible. So usually on, turns off. In the presence of the product. Again, this is the product of the reaction enabled by the enzyme. And in our simplified examples, previously we looked at the substrate as a food item that needs to be broken down. In the repressible example, we're going to look at the product as a, let's say, a raw material that needs to be made. So we'll see an example with an amino acid. So if that product is made and it is in high demand, then it's going to keep flying off the shelves, right? So if you make glycine amino acid and proteins are constantly being made, then it's a good idea for the cell to keep making amino acids because they're being used, right? So just like in economics, right? If you're supplying something in a warehouse, if it is popular, people are buying it, then it really serves you to keep making that. But let's say this doesn't happen. If you get a low demand for the product that you've made, then the product builds up, accumulates. And you get more product than you need, then this excess of product, I'll just put it in here, excess product becomes a signal. Let's see. I'll say it serves as the signal. Serves as a signal to stop making more. All right, so we go over here. Building up, building up. So again, as long as that diamond molecule is being used by the cell, it gets taken away from where it's made and put to use, then it's a good idea to keep transcribing. But when there's too much of this, then the excess, some of those molecules go back and interact with the transcription factors. So they interact with the activator or they interact with the repressor and they cause those to stop transcription. So now we have on the left an active repressor stops transcription. On the right, an inactive activator also stops transcription. And then your genes complain that they're being repressed. All right, I know I've been talking a whole bunch, so I'll give you a chance to weigh in here. Nice little 50-50 question. So this diagram here, in the presence of the purple circle molecule, does that cause the gene to be induced or repressed? I don't know how many people are out there. All right, and I got one. Anthony, I was looking for participation on the poll here. So majority is coming through as induced, and that'll be the right answer. So I use the term usually, but I guess you can also think of this as in the absence of that signal molecule, transcription is off. Okay, thanks, Anthony. Good to know. Who knows what kind of connectivity everybody has. Okay. So in the absence of the signal molecule, transcription is off, and it is caused to be turned on by the signal molecule. So that makes it inducible. It's being induced. I got to change the colors on these. This one, it comes away too easy because the color coding the book uses is green for activator and red for repressor. So I'll just give you the answer to this one. But we'll talk about why it's an activator maybe. So this is an activator molecule because when it attaches to the DNA, that causes transcription to happen. So up here, it's not able to attach to DNA. That does not allow transcription to happen. But in the bottom here, when it interacts with the substrate, then it changes shape, can interact with DNA, does allow transcription to happen. So I'll just spell those out here in the notes. So an activator allows transcription to happen when it attaches to DNA. So when the activator attaches to the DNA, that enables transcription to happen, and the repressor is really the opposite kind of molecule. Prevents transcription from happening. Shorthand this. Prevents transcription when it attaches to DNA. So in both of these cases, this when it attaches to DNA, that's the important part. Because we can see that a repressor, in the repressor examples, if the repressor can't attach to DNA, then it doesn't prevent transcription. So it's still a repressor, but it's not doing its job. So kind of its job is to prevent transcription from happening. And the activator has the job of allowing transcription to happen. All right. Questions before we move on from that? So we covered some basics about the process. This happens over quite a few different genes in bacteria. And then we'll talk about a specific example here. And this is a very common example. You might have seen it before in other classes. This is the process of digesting lactose in bacteria. So you don't need to know the fine details of this. Lactose is a disaccharide sugar, so it's made up of two sugar building blocks, galactose and glucose. And when bacteria cells encounter lactose, they can respond to that by making the enzymes that break it down. So lactose is kind of a rare sugar to encounter. We talked previously about lactose intolerance. When people are lactose intolerant, their body is unable to digest lactose, and the symptoms you get from eating dairy products are basically that the dairy products, the lactose from that goes into your digestive system without you breaking it down, and it becomes a food source for bacteria. And they kind of go nuts and break that down, and you experience negative effects from your bacteria breaking down a lot of sugar in your digestive system. So it's not a sugar that bacteria often run into. It's known as milk sugar. I don't know where else it's found. It's not unique to milk as far as I know. But when these bacteria encounter it, then they have to turn on the ability to digest lactose, because they don't usually have that on. So the ability to digest lactose involves, as this slide says, getting it into the cell using this permease transport molecule, and then breaking it down using this long-worded enzyme named beta-galactosidase. So beta-galactosidase converts lactose into its simpler sugar building blocks, and those can be digested by the more commonly expressed genes that break down simple sugars. Okay, so we got the, as I said, permease enzyme or transport molecule, and then beta-galactosidase. And so for those, there would be genes for those, and to make those enzymes and transport molecules, the genes for those have to be expressed, have to be transcribed and translated, right? And because this is a bacterial system, we're going to look at a bacterial, what's called an operon. So it's a little more than just a gene, and it relates to something we saw previously with bacteria, these polycistronic regions. And it's a long word, but it basically means, as you saw, 1, 2, 3, or multiple genes that all get transcribed together. I was a little torn as to whether to ask you these questions. I think I'll probably give you the answer to this one. We're running a little low on time. So as a polycistronic region, there's this one promoter region. So we got promoter regions, both colored the same, this kind of goldenrod color. The promoter here is a single promoter that causes the transcription through these 3 genes. So this is a polycistronic feature. So an operon the operon is a feature we see in bacteria that's not only these genes that are expressed together. So we saw an example of 3 genes all present in the same operon. They'll all get transcribed at the same time. They'll all get translated from that RNA piece. And it's not accidental how those 3 genes end up in the same operon. They have a similar or related function. So in the Lac Operon, the genes that are transcribed here are responsible together for breaking down lactose. So by transcribing these genes, and the specifics of this, I don't dwell on the specifics too much. So the names for the genes, they're called LAC-Z, LAC-Y, LAC-A. I don't need you to know the names for them. If I ever give you the names, I'll also give you a diagram for what they're doing. But it does help to show what these genes are containing the instructions for. So they're color-coded. The LAC-Z gene makes the beta-galactosidase enzyme that was responsible for breaking down lactose directly. LAC-Y makes that permease molecule that allowed lactose to get into the cell. And then LAC-A, transacetylase, not fully understood as last time I checked into it. But these are related functions, right? So to make use of lactose, the cell has to get it into the cell and break it down. There are separate genes for that. And with the Operon system, the bacteria can turn both kinds of genes on at the same time. All right, so then I wanted to also show you that in a little more diagrammatic fashion. All right, so promoter is what? LAC-Z, LAC-Y, and LAC-A. So transcription happens, then you would get the RNA molecule from the DNA starting point, and that RNA molecule contains the LAC-Z RNA, the LAC-Y RNA, the LAC-A RNA. And so in there would be start codon, stop codon for each of these. So if we went from that to translation, then the LAC-Z coding region produces this polypeptide, which is the beta-galactosidase. The LAC-Y translation gets us the permease, and so on. So the polycistronic benefit is that there's one instance of transcription, and then each of these are independently translated into their respective proteins, and they can go on to do their respective jobs. And then maybe one more thing right before we're out of time. What we'll be spending a little bit of time on is the expression, the regulation of the expression of this operon. So RNA polymerase would love to get in here, recognize the promoter, and transcribe these genes. Right now, it's being prevented from doing that because there's the repressor molecule that is attached, bound to the promoter region. So this repressor molecule, again, is made by a gene for the repressor molecule, and in its active form, it prevents transcription. So we'll see scenarios that are going to affect how this operon is transcribed, whether it gets transcribed, what kind of products it gets made, and kind of walks through the process of discovering how all these independent pieces fit together in the central dogma as it relates to the operon. So, questions? That is our time for the day. If there are no questions, I will get assignment 7 up. I'll give you a week and then maybe even more to finish that up. It'll be related to this, and maybe a little bit more that we haven't covered just yet. I'll have to see what ends up on the assignment. So it'll be up on Canvas pretty soon. Really just looking to have you get that done before we part ways right before the final exam. No more questions? Okay. Well, have a good day. We'll see you on Thursday.